And here we are, ladies and gentlemen, boys and girls, children of all ages, our final lecture for Chapter 3. Our final macromolecular group is the nucleic acids. Nucleic acids, this is the genetic material, the DNA, RNA, the means of storing the information on how to make and maintain you. DNA, RNA, deoxyribonucleic acid, ribonucleic acid. They're both based on a sugar that may or may not have a hydroxyl group. Deoxy, no hydroxyl. Ribonucleic has the hydroxyl. Now, when it comes to the nucleic acids, they're going to be found in multiple places throughout the cells. In eukaryotic cells, eukaryotes, remember, are going to be organisms that have a nucleus in their cell. So, for us eukaryotes, you're going to find it in the nucleus. You're going to find it in the mitochondria. You're going to find it in the chloroplasts. For prokaryotic cells, these are going to be the bacteria, the archaea, the single-cell things. Well, no internal membranes, no internal organelles means the nucleic acid, the RNAs, the DNAs are just going to be free floating throughout the cell. Now, you've probably heard the term genome. Genome is the collection of all your genetic content. Most of the time when people refer to the genome, they're referring to just the DNA, the double helix. But you got to be careful there. For all of us eukaryotes, yes, genome is that. But when you start getting into the microbiology, you start to see that there are things that don't use DNA. These are going to be viruses. But for the most part, biologists, when they say genome, they are referring to the DNA, the sum total of the DNA in each of your cells, the complete blueprint on how to make and maintain you. Now you'll see that the genome is packaged in what is known as chromatin and chromosomes. Turns out the double helix that you've heard about since you were a small child. Turns out that double helix, the two strands of DNA that wrap around each other, while stable, still can be broken relatively easily, still can be damaged. So, over hundreds of millions of years of evolution, we eukaryotes have evolved to take that long double strand and wrap it around small proteins. Kind of like how when you go to the store and you buy sewing thread. It's not just in a bag, because then it'll get all tangled up and knotted. No, it's wrapped around a thimble. Keep it nice, keep it safe, keep it from being knotted up, torn. Well, that's what chromatin is. Taking that double helix, wrapping it around histones that are acting like thimbles. Well, you take that chromatin and you start wrapping it around bigger and larger proteins to really package it down, and you get what's referred to as chromosomes. You've seen on TV and everything the 23andMe, how to get your DNA tested. The 23andMe, well, basically that means that, that's referring to the fact that you have two sets of 23 chromosomes. Two sets. One complete set, 23, chromosomes 1 through 23 from mommy. The other set, chromosomes 1 through 23, you got from daddy. Now, when it comes to the DNA, your double helix, there's going to be thousands of genes, 10,000 genes there. Each gene is a different instructions on when and how to make a protein, a polypeptide. Now here's the thing though. Your genome is so packaged away as chromatin chromosome, it's hard to get at. It's hard to do stuff with. And you want it that way because you want to limit the chances for damage. So you have to have ways to get at those genes, get at the information, the blueprint. Well, we have it. It's called RNA. Messenger RNA is a copy of one or more genes, one or more instructions. Think about it. If you went to the library has a book that has information you need. That book has 10,000 pages. That's going to be one big, heavy book to carry. You could check out that book and carry it around with you, carry it to the dorms or home, or you can go find the nearest Xerox machine. Make a copy of the one page of information you need. That copy, that you can just fold up, put in your pocket, walk away, no big deal. Leave the 10,000 page book there. Well, that's basically what your cells do. The DNA is the 10,000 gene, 10,000 page book. The copy of the one gene is what messenger RNA is. Messenger RNA is a throwaway copy. It's got the information you need. Your cell will read it. Cell will make what it said the instructions say to make, and then the messenger RNA falls apart, gets broken back down. The nucleic acid, recycled. The copying of the information, that single gene or multiple genes worth of information, depending on what organism you're talking about, that's referred to as transcription. Transcription. You talk to somebody who's a transcriptionist. All they do is they copy down word for word what is said in a meeting. That's what transcription does. Copies down DNA base by RNA DNA base, makes a complete copy, but does it in RNA. Transcription. Now, other things are going on in the cell that, also needs nucleic acid. We're going to see in later chapters when we talk. Translation. The reading of the information on the messenger RNA to make the polypeptide, the primary structure. Well, you're going to need transfer RNAs. Transfer RNAs are involved in translation. They escort in the amino acid, one after another. And then, as I talked about before, the ribosomes. The ribosomes actually make the polypeptide. They make the peptide linkage between amino acids. Now, DNA and RNA, its monomers are referred to as nucleotides. Every nucleotide has three components: a nitrogenous base, as we see up here, the nitrogenous base. Why do they call it nitrogenous base? Because somewhere in there is going to be a nitrogen instead of a carbon in one of the cyclic rings. A pentose sugar, right here. Right here. Pentose sugars. Hydrogen, hydroxyl. Base for DNA, base for RNA. And then finally, one or more phosphate groups. There you go, our functional groups again. That's why I told you got to learn them. The sugar, the pentose, hydrogen, hydroxyl, deoxy or ribose, the phosphate group, and then here is our pentoses. So, how's this work? Well, notice cytosine, thymine, guanine, adenine. These are our DNA nitrogenous bases. Uracil subs in for thymine in RNA. This is how our cells, all living cells, can tell the difference between DNA or RNA. Is there hydroxyl, yay, nay, and is uracil present? The ribbon you see here is one strand of DNA. The other ribbon is another strand. They wrap around each other in what is referred to as a helix pattern. Think of it as a ladder. You take a ladder, you're not twisting the ladder. What you're doing is you're taking the ladder and you're wrapping it around a pole. The rungs of the ladder are where the nitrogenous rings are. They form the rungs. The uprights, well, that's the sugar and phosphate strung together. When you look at the rungs between the sugar phosphate strands, thymine, adenine, guanine, cytosine, they form the rungs via hydrogen bonds. Remember back in chapter 2 I told you hydrogen bonds are extremely weak. They're easy to make, they're easy to pull apart unless you have billions of them. That's what holds the two strands of the DNA together as the double helix. Billions of these between the nitrogenous rings. Thymine, A to T has 2 bonds, G to C, 3 bonds. Now, the individual nucleic acids, the nucleotides, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. They're held together from the oxygen of the phosphate group to the 3rd carbon of the ribose, or the deoxyribose, if you're talking DNA. This phosphate, sugar, phosphate, referred to as a phosphodiester bond. 4 macromolecule groups, 4 bonds we have now talked about. The phosphodiester, phosphate, sugar, phosphate, sugar. You'll see some people refer to this as the sugar-phosphate backbone and the nitrogenous bases pointing in towards the center. Now, as I mentioned, DNA is too big and bulky. It's wrapped around proteins. It's wrapped around itself, forming chromatin and chromosomes. It's big, it's bulky, and you don't want to mess with it. So you're going to make a copy. That copy is the RNA. The RNA for us eukaryotes is only going to be 1 gene long. For the prokaryotes, the bacteria, the archaea, it can be 1 to 5 genes all together on one mRNA. It's not a direct copy. It's not CA at, put A. What you get, though, is its opposite. Wherever you see an A in the RNA, you're going to see U's. Remember, there's no T's. T's are only in DNA. Every time there's a T in the DNA, A's will be placed in the RNA. GC, its opposite, its complement, CG, and so on. So it's the mirror image of the DNA strand. Messenger RNA is made. If it's in a eukaryotic cell, that messenger RNA will then travel out of the nucleus. If it's a prokaryotic cell, well, it's made and it's just there in the cell. It's going to be bound by ribosome or rRNA. rRNA, the ribosomes we're going to see later chapter, are composed of 2 subunits, large, small. They will sandwich the messenger RNA. Doesn't matter if it's prokaryote, eukaryote. They will bind to it and they will go codon by codon. A codon is 3 nucleotides. 3 nucleotides, 3 nucleotides, 3 nucleotides. They will read down every 3. Every 3 calls for a different amino acid. That amino acid is going to be chaperoned in by tRNA. Every codon present will have a specific tRNA that goes with it. How that works, we'll talk about in a later chapter. But the ribosome not only sets what's the codons, but then once the correct tRNA with the correct amino acid comes in, it synthesizes a dehydration reaction. It adds one more amino acid to the growing polypeptide. You see here, 3 by 3 by 3, codon by codon by codon. The large, the small subunit moving along. In comes a new tRNA with the correct, which corresponds to that codon, brings in another amino acid. That amino acid gets added to the growing polypeptide, and the tRNA gets kicked out. DNA, RNA, different structure, different functions. Different sugars, different nitrogenous bases sometimes. Different structure, different functions for all living cells.